Hi everybody, and welcome back to our monthly lab seminar. So glad everyone could make it today. We're going to start off with the seminar with a 24-year-old patient that presented to her doctor with severe abdominal pain, chills, and sweating. Due to the symptoms presented with, a stool, midstream urine, and blood tests were collected. It appears our patient is suffering from symptoms that are usually associated with gastrointestinal tract infections. These symptoms can occur due to bacterial, viral, or parasitic infections. Along with our patient's symptoms, vomiting, diarrhea, and fever are also associated with GIT infections. These symptoms may be related to an infection at the mucosal surface of the GIT, direct microbial infiltration of the gut, as well as microbial toxins.
This patient displayed relatively common signs and symptoms for many gastrointestinal tract illnesses stemming from bacteria, viruses, and even fungi, along with potential injuries. Therefore, because of this, it was imperative to obtain and interpret the correct results from the cultures. As expected with these symptoms, there was no growth in the blood. There are, however, two pieces from the data that strike as significant, one being somewhat expected with the symptoms present, while the other being unusual. What struck as unusual with this 24-year-old patient was the lack of isolated pathogens from the cultures in both stool and lack of growth in midstream urine. Given the significant signs and symptoms, this was unusual with the types of agar used. Beginning with the stool sample, while the wet film does not provide the necessary data to identify the pathogen, with the symptoms present and the positive reading for red blood cells and white blood cells, it is clearly an indication of a pathogen or injury in the gastrointestinal tract. Red blood cells and white blood cells do not normally appear in stool, and therefore their presence is an indication of something abnormal. The wet film unfortunately does not provide enough indication on what type of pathogen it is, or whether it is a pathogen at all, as blood in stool may also be an indication of injury or conditions such as ulcerative colitis. While the McVean agar has some use in diagnosing specific gastrointestinal tract diseases, the lack of pathogens from the McVean plate is to be expected, as McVean agar, although selective and differential, is commonly used for enterobacteria already in the gastrointestinal tract, such as Escherichia coli and Enterococcus species. Therefore, it is unlikely for this agar to provide us with a clear result. Following on with the selenite broth, a selective medium used for isolating salmonella, while simultaneously reducing the growth of normal fecal and gastrointestinal tract flora. Therefore, these results, with being negative, indicate that salmonella poisoning can be ruled out. Similarly, as an XLD plate is selective for Salmonella and Shigella species, we can rule these out too, as there was no significant pathogenic growth. This, however, is only a small part of the numerous pathogens that may be causing the symptoms this patient is receiving. However, the use of XLD plate is important, as many Enterobacteriaceae, both normal flora and pathogenic, can still grow in the XLD plate. Thus, this possibly rules out a further number of potential pathogens. The lack of pathogenic culture on horse blood agar is unusual given the symptoms present, and given that blood agar is an enriched medium with many organisms able to grow in it. However, the lack of growth is ironically beneficial in this regard, as it rules out many pathogenic bacteria. As there was no growth aerobically, a reasonable assumption can be made that this bacteria is an obligate anaerobe or grows under microaerophilic conditions with CO2. These symptoms and growth from the stool sample, or lack of growth, as well as the wet film, combined with the lack of anything significant in both the urine plates and the wet film's microscopy, indicate that this person is most likely suffering from a gastrointestinal tract infection.
The patient presented with many common symptoms of a GIT illness. However, the lack of pathogenic growth eliminates many common bacteria associated with these symptoms. XLD is commonly used to isolate Salmonella and Shigella, and with this plate presenting no suspicious colonies, it can be assumed it is neither Salmonella or Shigella causing the patient's illness. Bacteria such as Klebsiella species can be ruled out due to the lack of pathogenic growth on the McVean agar plate. Even though there is no pathogenic growth visible, this in itself provides some insight into the potential cause of the patient's illness. A viral or parasitic infection should be considered, as they have the potential to cause such illnesses and would not present on agar. Due to the lack of growth, a bacteria such as Micrococcus should be considered, as they present with zero growth on any plate. Fastidious bacteria such as Fastidious E. coli could cause such health problems while also not showing up on the plates used. Enriched agar such as a chocolate agar plate would be able to determine if this bacteria was present. While Enterococcus faecalis is normal flora of the gut, if the digestive tract is damaged, it has the potential to cause serious infection. These are serious infections due to their common resistance to antibiotics. A mannitol salt agar plate would allow for growth if Enterococcus faecalis was present. Another bacteria that should be considered is Clostridium, specifically Clostridium perfringens. Clostridium perfringens is a very common cause of food poisoning, which aligns with the symptoms that the patient is suffering from. In order to determine whether Clostridium perfringens is the cause of this illness, a stool culture test can be done to determine the number of bacteria in the feces. An egg yolk media plate can also be used.
The methods conducted to identify the possible cause of the symptoms included a wet slide, blood, stool, and urine cultures, as well as microscopy. On day 0, 3 samples were obtained: a stool sample, a blood sample, and a midstream urine sample. The stool sample was inoculated and streaked onto horseblood agar, McConkey, and XLD agar media using an aseptic technique in order to identify any pathogenic bacteria present. The plates were incubated for 24 to 48 hours at 37 degrees Celsius in aerobic conditions. These media were used to identify possible Staphylococcus, Micrococcus, E. coli, Enterococcus, Salmonella, and Shigella infection. A cellulite broth was also set up by inoculating the broth and incubating it at 37 degrees Celsius for 24 hours. A wet slide was set up and viewed under the light microscope at 100 times objective, where white and red blood cells were observed in equal amounts of 2+, but no suspicious organisms were observed. The presence of white and red blood cells in the stool are an indication of infection. A blood culture was set up using 2 bottles, one incubated anaerobically and one aerobically, for 5 days at 37 degrees Celsius. This was conducted to identify if any pathogens are present in the blood, which is usually sterile. A midstream urine sample was cultured on horseblood agar, McConkey, and orientation chromogenic agar, and microscopy was completed. The microscopy indicated the presence of red and white blood cells, as well as epithelial cells at less than 10 times 10 to the power of 6 per litre. The lack of epithelial cells indicates a good quality sample. On day 1, the agar plates were observed, and they showed no pathogenic growth on any of the plates. The cellulite broth was also negative for any pathogenic growth. There was also no significant growth for the urine culture. On day 4, the blood cultures were observed and no growth occurred. Further testing is required to identify a possible cause of the symptoms, including incubating the stool sample cultures on HbA anaerobically, the McConkey agar in microaerophilic conditions at 42 degrees Celsius, and setting up a CCDA agar plate to incubate in CO2-rich conditions. This would identify Campylobacter and any strict anaerobes, which are the likely cause of infection, following investigation of the case notes and initial results. The stool sample could also be sent off for further testing using microplex PCR, which could not be completed in the microbiology lab.
To refresh our memory, let's run through the process. Don't forget to catalogue the specimens as soon as they arrive. Unknown specimens impact patient care and are really not a good thing. If we need to send some of the sample off for a multiplex PCR, we would do that now. Immediately, we record our microscopy, quantitate the specimen, and describe it in detail. Then our microscopy. In the case of the stool sample, we would perform a wet film to look for parasites such as Giardia or Cryptosporidium. Don't forget to note the red cell and white cell presence. From here, we culture the specimen on a Campylobacter plate at 42 degrees in microaerophilic conditions. Otherwise, the organism won't grow, which is why we didn't see it in the plates mentioned previously. On day 1, we ID the specimen from culture, gram stain, catalase, and oxidase. Further testing for identification includes the hippurate test, nalidixic acid, and cephalothin discs. We'd also need to set up a Campylobacter UCAST for the antibiotic profile. Here is the UCAST guideline, valid from the start of this year. If you would like a copy, we can provide you with one. And here is how you would set up the plate. This will also be attached to the official document. On day 2, we record the results of our final identification and write the doctor's report ready to be sent off immediately. In conclusion, the initial plates did not assist in the diagnosis of the female patient. Whilst it was appropriate to conduct the array of tests, the neglect of a routine Campylobacter plate was a mistake and should be avoided in the future. The lack of pathogenic growth did indicate which tests to pursue for further diagnosis. If you are unsure of the correct procedure to follow for certain specimens, ask a supervisor.